Hello everyone, welcome to the IDLS Academy. Today we are going to discuss the topic Introduction to Molecular Biology. So first of all, we are going to see that what is the molecular biology. See, molecular biology is simply what we are going to study in this microbiology uh, about the structure, composition, and interaction of molecules and macromolecules. Okay, what we are going to discuss? We are going to discuss the structure, composition, and interaction of molecules and macromolecules. Okay, macromolecules which are essential for the biological process. Okay, so the macromolecules and the molecules which are the essential for the biological process, their structure, composition, and interaction we are going to study in molecular biology. Macromolecules means that about the carbohydrate, about the protein, about the fats. Okay, so that's all things that we are going to discuss in molecular biology. And this molecular biology is totally focused on understanding that the molecular basis of biological activity, including their mechanisms and interaction of molecules in and between the cell, the mechanism which are conducted within the cell or between the cell, and even the interaction of the molecules within the cell and between the cells. This microbiology are very important for the understanding how the molecules make up the cell, determine the behavior of living things. We all know that cells are what? Cells are the smallest unit of the living beings. So how these cells are made and how what is the behavior of this cell? Okay, how this cell are made and what is the behavior of this cell? We are study in molecular biology. Okay, for example, it's integral to understanding vascular disease process which are influenced by a single gene defect. Okay, so there is a defect which is called a vascular disease defect, and this defect is caused only when a single cell defect are occur, and this defect are occur due to the multiple genes product interact with the outside the environmental factor. When the multiple genes product interact with the environmental factor, then there is a defect arises in their single gene. Okay, now this defect in a single gene caused a vascular disease process. Okay, so come to the next slide. That is your eukaryotic cells. See, we all know that all the living beings are made up of what? They have what? They have a eukaryotic cells. Okay, living organisms. Now these eukaryotic cells have different organelles are present, like your mitochondria. Okay, then your peroxisomes, lysosomes, microtubules, plasma membrane, centriole, smooth endoplasmic reticulum, microfilament, ribosomes, rough endoplasmic reticulum, nuclear envelope, nucleus and nucleolus, and their Golgi apparatus. See, this part is what? This is your mitochondria, and we all know that mitochondria is generate the heat, generate the energy. Okay, because it is called a powerhouse of the cell. This is very basic thing, and it also used for a calcium storage. Okay, this mitochondria. This is the function of the mitochondria. Now this Golgi apparatus is used for a protein synthesis, lipid synthesis, and polysaccharide synthesis. Polysaccharide synthesis. So this Golgi apparatus have three functions. Either they are used in a protein synthesis, either they are used in a polysaccharide synthesis, and either they are used in a lipid synthesis. Now next, use your peroxisome. Peroxisome is used for a digestion. This structure is what? This is your peroxisome. They help in what? Digestion. Okay, like your apoptosis, when the cell is damaged beyond a repair. It also help in a apoptosis. Apoptosis. Okay, which means that when a cells are damaged, then they repair themselves. Okay, when the cells damage, then they repair beyond. This is the function of the peroxisome. The next is you. Sorry. This is the function of your lysosomes, not peroxisomes. There is a little confusion. Okay. So this function is what about the lysosomes? They help in digestion and they help in a apoptosis. Peroxisome is help in a detoxification. Okay. They help in what? Detoxification and lipid metabolisms. Lipid metabolism. Okay, the next one is your microtubule. Microtubule help in a locomotion. This structure is your microtubule, and this structure is your lysosomes. Okay, microtubule help in locomotion, and it also gives the shape to the cell. Okay, now the next is your plasma membrane. Plasma membrane is, this membrane is called your plasma membrane. Okay, this area, and this plasma membrane is used for a protection, regulation, and transportation. Okay, protection, the main function of the plasma membrane, regulation, and transportation. This centriole is help in a cell division. This structure is what? This is your centriole, and they help in a cell division. Now we have two type of reticulum, that is your rough endoplasmic reticulum and smooth endoplasmic reticulum, and they are what? They are help in a protein synthesis. Protein synthesis, both this rough endoplasmic reticulum and this rough and smooth endoplasmic reticulum, protein synthesis. And your ribosomes is used for a protein synthesis. Okay, now the next one is your microfilament. Microfilament is used for a cell support, and they are also help in providing the structure. Provide the structure. So this is two main function of the microfilament. Then nucleus obviously nucleus is for storage of the DNA. This structure is what? This is your nucleus. Nucleus is for storage of DNA. Okay, and nuclear envelope separate the nucleus from a cytoplasm. This is what? This is your cytoplasm, and this is your nuclear envelope. This nuclear envelope separate the nucleus from cytoplasm. Okay, now nucleolus is used for a ribosome production. Ribosome production. So these all about the part of the eukaryotic cells and their function. Now come to the next slide. DNA. We all know that DNA are present in where? They are present in a nucleus. Okay. They have 22 chromosomes. Okay. And they are in pair and plus 2 sex chromosomes, which determine that the sex of the living things. Either they are male or either they are female. Okay. 2 copies of each are present, and in human being, we around 99.9% DNA are identical. DNA are identical. Okay. So we have only 0.1% of different DNA from each human being. Okay. So we are identical around 99.9%, and this 0.1% give us a unique characteristic. And if we talk about the chimpanzee, then they have a 98% identical. Around 2 m DNA enough to travel to sun and back 600 times. Okay. If we take around 2 meter of DNA, then the length of the DNA is like that. They travel to the sun and back around 600 times. That's the length of the DNA. Therefore they are what? They are tightly packed to each other. Now this is your structure of the DNA. This is your sugar phosphate. Okay, sugar phosphate, and they are what? They are a backbone. This colorful thing is what? They are a base pair. This red, this blue. Okay. Now this red one is your adenine. Okay. This blue one is your thymine. This structure is your guanine, and this structure is your cytosine. Uh, brown is what? Brown is your guanine, and this green is what? Cytosine. And this is your nitrogenous bases. Okay. Now we all know that the thymine made always bond with adenine, and this guanine always form a bond with a cytosine. See, this is the what? This is base pair which are arranged. One is arranged from 5 prime to 3 prime, and another one is here arranged 3 prime to 5 prime. They are always arranged like this. Okay, suppose this is your structure of the DNA. So if it is 5 prime, then this end will be a 3 prime, and this will be a 3 prime, and this will be a 5 prime. So this is the structure of the DNA. This is what? This is the structure of the DNA. Now come to the next. DNA function. Okay. The DNA function is what? It give a blueprint for life, and it is used for a duplication of the new cells and make a protein for a biological function. What happened? This is your double stranded DNA. Then this double stranded DNA are transcripted and converted into a messenger RNA. They are what? They are a double stranded DNA. When they are transcripted, okay, now this band is converted into a single strand. It is called a mRNA. This mRNA again, this mRNA go into a ribosomes. Okay, and this ribosomes convert this mRNA into a protein, and this process is called what? They are called a translation. Okay, so the DNA, what? DNA is help in a production of the protein, which are help for a biological function. And we all know that when this DNA are duplicate, they form a new cells, and they are also used as a blueprint for a life. Now this is the structure of the DNA. DNA have exons, start site, and control site. Okay, this is your structure of DNA. Sorry, gene. Now mRNA. mRNA what? DNA are grouped into a three codons. Okay, they have start and stop codon. mRNA after that they modify and travel out of the nucleus. This is your mRNA splicing. See, this is your gene. This is your exon 1, exon 2, exon 3. This is your antron, intron, and intron. Okay. When this transcription occurred, transcription occurred, then the RNA synthesized, then nuclear RNA, then after that RNA splicing occur. This intron is removed and only exons are there, and this is called what? This is called a messenger RNA. Alternative of the splicing. Now translation. Translation is what in translation mRNA, which is called a messenger RNA, are converted into a protein. Okay, and this all are occur in a ribosomes. They are occur what? They are occur in a ribosome. One codon, one codon are equal to a one amino acids. Okay, we all know that this protein are made up what? They are made up of a amino acid. So one codons are equal to a one amino acid. Now protein. Protein what? Protein are a huge proportion of a cell. After water, if we say that the huge proportion of what things are present after water, then the answer is what? Protein. The main function of the protein are what? They give us structure of the cell. They produce enzyme, transmembrane receptors, and hormones. There are four level of structure. We all know that primary, secondary, tertiary, and quaternary. That we are going to discuss in the next slide. So this is the structure of the protein. First structure is what? They are primary structure. Here what? There is a single sequence of amino acid. Okay, in a sequence of in a sequence of chain of amino acid. Now the next one is your secondary structure. This structure is occur when the amino acid are linked by a hydrogen bond. Suppose this is your single type of amino acid sequence, and this is a single type of amino acid. When they are form a bond with the OH group, then this structure is converted into a secondary structure. Then the tertiary structure, tertiary structure is occur when certain attraction are present between the alpha helix and a plated sheet. Okay, alpha helix and a plated sheets. Now then last structure is your quaternary structure. This structure of protein is consists more than one type of amino acids sequence. Now protein interaction. Protein can interact with either they are interact with protein. Okay, either they interact with mRNA, either they interact with the DNA. And when these proteins can form a bond with each other, and these are totally depend upon their structures and their charges. Okay, this is your gene expression that we already discussed that this is your DNA transcription occurred, then a primary RNA transcripted RNA process, then RNA are formed. This RNA again translation control protein are formed, then this protein are inactivate and inactivate active proteins are formed. Now come to the next slide. Common terminology that we use, that is your genome, genotype, pseudogenes, novel protein, putative gene, local ally, chromosomes, region, dominant, recessive, homologous, symbol, cDNA, motif, Dalton D or KDA. Common abbreviation that we use, EST. The first abbreviation is your EST. That means that express sequence tag. SNP, that is your single nucleotide polymorphism. ORF, that is your open reading frame. Okay, this is what? This is your open reading frame. UTR, untra, un, untranscript region. So experimental techniques like cut, change, knockout genes, transgenics, sequence genes, protein, express protein, micro errors. Future of this molecular biology. personalized medicines. They are also used in a target specific drugs like adipose tissues, gene therapy, competitive genomic. Okay, so this is all about the future of molecular biology. Thank you for the watching. Please like, share, and subscribe our channel. If you have any query, just let us know in a comment box, and you can also send your query to the helpline number, which are also mentioned in your slide. You can also follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn. You can also get our app from Google Play. Please subscribe the channel ITLS Academy in YouTube. And if you have any query, just let us know in a comment box.